everyone, and welcome to another rendition of Global IMMUNOTALKs. It is a pleasure hosting Global IMMUNOTALKs, but this one is a spectacular one for me because I'm hosting one of my favorite immunologists. I have the pleasure to introduce today Lucy Walker, who I have met and I found out about 18 years ago and seen her on a regular basis. And I have to say, Lucy is, as a speaker, as you will find out in a bit, is a real treat. So I'm very happy to have you here, Lucy. A bit about Lucy Walker. She studied biology in Nottingham and then immunology at the University of Bath, both in the UK. And then she did a postdoc with Peter Lane at the University of Birmingham, followed by a traveling fellowship, which is like an advanced postdoc at UCSF with Abu Labas. In 2004, Lucy came back to the UK and became a senior lecturer at the University of Birmingham. And she was then promoted to a reader. This is a UK thing. She became promoted to a reader at the University of Birmingham in the division of immunity and infection. And in 2013, she moved to London, to the UCL, University College of London, where she is a professor of immune regulation at the Institute of Immunity and Transplantation. Why am I so excited about Lucy Walker? So her work is actually, Lucy is interested in how T-regs work and how they actually regulate immune responses. And her early work is in the area of T and B cell collaboration, and she led to the discovery of how CD28 co-stimulation is needed for the development of follicular helper T cells. And she subsequently identified the role for CTLA-4 in regulating follicular helper T cells. But one of the things that shining super paper was basically the discovery that T-regs are not anergic in vivo, but instead proliferate in responses of antigens. And then she showed that they use CTLA-4, the inhibitory protein CTLA-4, through a very, very unusual mechanism involving the capture and degradation of the 7 molecules of co-stimulatory ligands, a process that's called Transendocytosis. If you haven't read that paper, you should. It's an amazing paper, super, super exciting. So Lucy obviously had a tremendous impact on the T-reg and autoimmunity world, and she won multiple awards and honors. But I would like to just mention one here as the Global Society Wolfson Research Merit Award, which is a big one. So, and you know, as Lucy, she's here now, as I told her earlier, what we do here at Global IMMUNOTALKs, we ask our guests a question and see what they have to say to this. And the question I would like to ask you, Lucy, is what is the advice you would like to give to your 25, 35-year-old self? And with this is my question for you. With this, I'll shut down my video and the stage is yours, Lucy. Thanks for being here.
Thank you. Well, can I start off by thanking you very much for the opportunity to be part of this fantastic seminar series. I think it's an amazing initiative and I've really enjoyed following many of the talks. So in answer to your question, Burkhard, I think I would say to the younger me, don't worry if the data that you're seeing sometimes doesn't make sense. It might just be that there's something that hasn't been discovered yet in your field that's needed to interpret it. And there have been a number of examples of this in my career. I remember being very irritated by background staining with a CD25 antibody when I was looking at T cell preps. And of course, this turned out to be regulatory T cells, but this was before CD25 positive T-regs had been discovered. And a lot of our CTLA-4 experiments also didn't really make much sense initially. And it's only when we learned more about the molecule and started to think about it in a different way that things really started to fall into place. So I used to be really troubled when I couldn't reconcile my data with the literature, but now I would say take note of these things, embrace them, hold on to them, and come back and look at them in a few years' time. And it may just be that they're a clue to something that we don't know about in the field yet.
Cool, thanks.
So now I would like to share my screen. How's that?
Looks great.
Perfect. Great. So what I'd like to talk about today is how CTLA-4 regulates autoimmunity. And just to set the scene, we know that our immune system needs to be tightly regulated, and the T-cell compartment in particular needs regulation because we need it to be able to respond very rapidly in order to protect us from infection. But we also need to avoid collateral damage, so T-cell responses against self-antigens that could cause autoimmune diseases. And one of the ways that we realized what a fine line the immune system has to tread here came from work done by Don Mason, who actually challenged the paradigm of one clonotype, one specificity, and argued that cross-reactivity is actually an essential feature of T-cell recognition. And in fact, he argued that you need a high level of TCR cross-reactivity in order to have the recognition capacity that we have with the number of T cells that actually fit in our body. So one implication of this cross-reactivity is that foreign reactive T cells and self-reactive T cells don't have a clear distinction. It's not possible to delete all of the self-reactive T cells in the thymus and just leave the foreign reactive T cells. So we don't do that, and pioneering work from the Sakaguchi lab in the mid-90s demonstrated that in fact self-reactive T cells don't all get deleted in the thymus, and they exist within our normal peripheral repertoire. If you take regulatory T cells away, these self-reactive T cells will cause autoimmune diseases. And then elegant studies from Mark Davis's group sometime later showed using tetramer staining that you can visualize these self-reactive T cells and quantify them in normal human beings. So we all have these T cells that are capable potentially of causing autoimmune disease. So how do we decide which T cells are allowed to become activated? And there's an important role here for accessory signals, so additional interactions between the T cell and antigen presenting cell that can provide context about the antigen engagement event. And I've just shown some of the accessory molecules here and selected therapeutics that target them. And arguably the key, the most important accessory signal is delivered through this co-stimulatory receptor CD28, which provides this important second signal for T cell activation, with the first signal being through the T cell receptor engaging peptide MHC complex. So whether CD28 is engaged or not is really important in determining whether there will be an immune response. So therefore, CD28 engagement needs to be tightly regulated. And one of the proteins that's involved in regulating CD28 is, of course, CTLA-4. So CTLA-4 binds to the same ligands as CD28, CD80 and CD86, B7-1 and B7-2, as they used to be called. But of course, CTLA-4 is inhibitory rather than co-stimulatory. Another key difference between CTLA-4 and CD28 is that CTLA-4 is an endocytic receptor. TCR signals will promote trafficking of CTLA-4 to the membrane, but it's continuously endocytosed, such that most of it is inside the cell at any one time. And you can see this if you just look at t-regs ex vivo and stain them for CTLA-4. So these are murine t-regs. And you can see there's actually very little surface CTLA-4 staining. even though when you do an intracellular stain, you can see that the cells are packed with CTLA-4 inside. But if you take those T-regs and put them at 37 degrees for a couple of hours and put an antibody in the medium to CTLA-4 or CTLA-4P antibody, this actually allows you to label the cycling pool of CTLA-4 antibodies. So these are CTLA-4 molecules. So these are receptors that are coming to the surface, binding to antibody and internalizing. So it's a highly dynamic receptor. So it's endocytic. It binds to its ligands with higher affinity than CD28. And with CD80, there's also an avidity advantage. And its importance in negatively regulating the immune system really was emphasized when Arlene Sharp and Tak Mack's groups generated CTLA-4 deficient mice. And these animals developed a lethal lymphoproliferative syndrome. We know, of course, that CTLA-4 is a very important molecule clinically. Anti-CTLA-4 antibodies are used to increase anti-tumor immunity, increase anti-tumor immune responses in cancer immunotherapy, and soluble CTLA-4 molecules are used to suppress immune responses in autoimmune settings. But despite the clear importance of this molecule from a clinical perspective, precisely how it functions has been hard to unravel. And one of the early clues to this came from experiments done by Manfred Kopf and Martin Bachmann, where they reconstituted RAG-deficient mice, so mice that didn't have T cells and B cells, with either wild-type bone marrow, CTLA-4 deficient bone marrow, or a one-to-one mixture of the two. The mice that received the wild-type bone marrow developed normal immune systems with a normal T cell population. The mice that received CTLA-4 knockout bone marrow generated immune systems with hyperactivated T cells, and these infiltrated tissues and caused disease. But the mice with the mixed bone marrow developed normal immune systems. There was no disease, and the CTLA-4 deficient cells in these animals didn't look any more activated than the CTLA-4 sufficient cells. So this experiment has been reproduced now by multiple labs. It's highly consistent. This is just our version of it here. So here, mice that received CTLA-4 deficient bone marrow have activated T cells with high Ki67, high ICOS, and low CD62L, compared to the recipients of wild-type bone marrow. And if we look in the bone marrow chimeric mice and gate just on the CTLA-4 deficient cells, we can see that these have a resting phenotype, essentially indistinguishable from the wild-type cells in the same animal. So cells that are expressing CTLA-4 are able to control CTLA-4 deficient cells in the same animal. And this sort of cell-extrinsic mechanism of action is, of course, very much in line with how regulatory T cells work. And there were experiments by Shimon Sakaguchi and Fiona Powry's group showing that T-regs express high amounts of CTLA-4 and use it for their suppressive function. And we were also doing these sorts of experiments at the time. We were using a mouse model of diabetes, where we were adoptively transferring over-specific T cells into mice expressing over in the pancreas under the control of the rat insulin promoter. And these T cells would destroy the insulin-producing cells, and the mice would all develop diabetes. But if we co-transferred antigen-specific T-regs, these would completely protect from diabetes. And so we asked whether those T-regs needed to express CTLA-4. And we got a very clear answer. The CTLA-4 sufficient Tregs could regulate diabetes beautifully, but the CTLA-4 deficient Tregs were completely unable to control disease. Even though we could see that they were surviving the adoptive transfer, they were getting to the right tissues, we could find them in the pancreas and so on. And one of the interesting things that we saw in this set of experiments is that CTLA-4 mediated suppression was associated with downregulation of the co-stimulatory ligands CD80 and CD86 on dendritic cells. So here these are murine dendritic cells, and you can see the decreased CD86 in the presence of Tregs. And in collaboration with David Sanson's group, we were able to provide a molecular mechanism for this, which is that CTLA-4 was able to remove its ligands from adjacent cells by a process of transendocytosis, so binding to them and removing them and internalizing them for degradation. And just to show you a couple of data slides on this. So here we're looking at human dendritic cells stained green for CD86. And you can see that after incubation with CTLA-4 positive Tregs, this green staining is largely lost from the surface of the dendritic cell, and instead you can find punctate staining within the Treg, and the captured ligands can be degraded. And importantly, these dendritic cells that have had their co-stimulatory ligands removed are now not able to co-stimulate T-cell proliferation through CD28. We could also demonstrate this transendocytosis process happening in vivo using mice, and we used mice that were expressing a GFP-tagged version of CD86. The wild-type Tregs that we'd shown would protect from diabetes, these were able to capture GFP-tagged CD86 in vivo, whereas the CTLA-4 deficient Tregs that couldn't control diabetes were not able to capture ligands. We can also measure this ligand transfer by FACS analysis. So here we're looking at Tregs from either human healthy donors or from individuals with heterozygous CTLA-4 deficiency. And if we look at the Tregs from healthy individuals, you can see that these are able to capture CD80 GFP from a donor cell in a manner that's CTLA-4 dependent. And these healthy Tregs are also able to elicit efficient CTLA-4 dependent suppression. So I won't go through all the controls here, but essentially the red bar is conventional CD25 negative T cells on their own. The blue bar is T cells where we've added in the Tregs. So here we're getting the suppression, and this is alleviated in the presence of anti-CTLA-4 antibodies. And Tregs from individuals with heterozygous CTLA-4 mutations show impaired ligand capture, impaired transendocytosis, and this is associated with impaired CTLA-4 dependent Treg suppression. And these individuals also had a very interesting immune dysregulation syndrome. And in fact, we can take patient CTLA-4 mutations and clone the CTLA-4 molecules, express them in cell lines, in CHO cells here, and examine their function, which aspects of their function are defective. On the left here, we've got some CHO cells expressing wild-type CTLA-4 in red, being incubated with CHO cells expressing CD80 in green. And if you leave these alone in the dish and come back a few hours later, you find that the red cells are now stuffed full of yellow vesicles. And this is where they've captured the green CD80 from the adjacent cells. Whereas on the right here, we've got CHO cells expressing one of our patient CTLA-4 mutations, and you can see that these cells are unable to perform transendocytosis, and it's really clear how much brighter the green staining is on the right than on the left. So we think that CTLA-4 is acting as a highly efficient molecular pump, capturing co-stimulatory ligands, removing them from antigen-presenting cells. And what this means is that this antigen-presenting cell is now not able to provide CD28 co-stimulation to T cells. And this mechanism explains how you can have a positive regulator and a negative regulator sharing the same ligands. It fits with this quite specific and unusual intracellular trafficking itinerary of CTLA-4. And it also fits with a cell-extrinsic role for CTLA-4 on Tregs. And if you're interested in the particular APCs that are targeted by CTLA-4 transendocytosis in vivo, I refer you to this paper by Vitaly Ovchinnikov's, who is a very talented PhD student in my lab. So the idea that CTLA-4 is functioning to regulate CD28 engagement also fits with experiments done by Al Singer's group some years ago now, where he showed that the disease phenotype of CTLA-4 deficient mice depends on CD28. And in fact, he was able to define the specific residues in the cytoplasmic tail of CD28 that you need to get the CTLA-4 knockout disease. So the biological role of CTLA-4 is to control CD28 engagement. And I'd like to switch gears a little bit at this point to talk about the role of CTLA-4 in t-reg homeostasis. Tregs are obviously expressing CTLA-4 at a high level. They're using it for their suppressive function. And one of the things we realized quite early on is that it was also having an impact on their homeostasis. When we took just normal wild-type mice and injected them with blocking CTLA-4 antibodies, we could see proliferation in the Treg compartment at early time points where there was no effect in the conventional T cell compartment. If we look in CTLA-4 deficient mice, these have increased Treg proliferation and an increased proportion of FOXP3 positive Tregs. And we subsequently showed that this was also the case in humans with heterozygous CTLA-4 deficiency, both when we examined the T cells direct ex vivo and after a brief in vitro stimulation. These individuals often have a higher percentage of Tregs. So given that CTLA-4 is operating to control CD28 engagement, its ability to control Treg homeostasis fits very well with work from Jeff Bluestone's lab over 20 years ago now, who demonstrated a key role for the CD28 pathway in controlling Treg homeostasis. So these data here are from NOD mice, the NOD diabetes model, and the decrease in Tregs in animals that lacked CD28 or the B7 ligands was able to explain the unexpected exacerbation of diabetes in these mice. Recent work from the Casanova group also indicates that this role for CD28 in Treg homeostasis also holds true in humans. Mutation of CD28 that prevents its surface expression is associated with a decreased Treg frequency. One of the ways that CD28 co-stimulation can be inhibited clinically is by using soluble CTLA-4 molecules, CTLA-4 immunoglobulin fusion proteins like abatacept. And this is because, as I said, CTLA-4 binds to the shared ligands with higher affinity than CD28, so this can act as a competitive inhibitor. And people with autoimmune diseases who've been treated with soluble CTLA-4 it often exhibits impaired T-reg homeostasis. So here, these are patients with arthritis who've been treated with abatacept, and you can see that after 3 months, there's a reduction in the frequency of FOXP3 positive T-regs. And then on the right here, this is a TrialNet study of abatacept in new onset diabetes. And again, you can see this decrease in the frequency of T-regs that's alleviated after treatment cessation. And we actually obtained some samples from this trial, and we could corroborate this decrease in the frequency of T-regs in recipients of abatacept, but not recipients of placebo. And we were particularly interested in looking at predictors of clinical response, and mostly in this study we focused on follicular helper T cells. But actually, if you look in the data set, T-regs also come up and are associated with the clinical response. So I think it's clear that targeting the CD28 pathway can have the detrimental consequence of impairing T-reg homeostasis. And obviously, if you're trying to treat an autoimmune disease and suppress immune responses, ideally you don't really want to decrease your T-regs. You'd like to maintain them and harness their immunosuppressive potential. As well as CD28, we know that IL-2 also plays a key role in T-reg homeostasis. Work from Shimon Sakaguchi's group showed many years ago that this spontaneous proliferation of T-regs in the steady state could be inhibited if you block IL-2. And Dan Campbell's group showed that T-regs use CCR7 to guide them to the T cell area of secondary lymphoid tissues where they can access IL-2 signals. And Ron Germain was able to visualize this process directly in vivo, identifying an IL-2 producing cell here in blue, surrounded by green FOXP3 positive T-regs that are also staining red for phospho-STAT5, implying that they're receiving IL-2 signals. So T-reg and conventional T cell responses are coupled via co-stimulatory ligands and IL-2. Activated T cells can use CD40 ligand to increase levels of CD80 and CD86 on APCs. These T cells also make IL-2, and both of these resources are used by the regulatory T cell as well as the conventional T cell. And so we became quite curious about how this relationship between CD28 signals and IL-2 signals operated in terms of maintaining T-regs. Do these pathways operate independently, or do they work together? And one of the first things that we did to try and address this was to ask whether IL-2 was able to augment T-reg proliferation in the absence of CD28 in CD28 knockout mice. So here we're treating mice with IL-2 complex, so this is IL-2, anti-IL-2, and when we put this into wild-type mice, we can see an increase in T-reg proliferation. Doesn't really affect the conventional T cells, and so this results in an increased percentage and absolute number of T-regs. And when we do this in CD28 knockout mice, we get a very similar picture. There are fewer T-regs to begin with in these animals, but IL-2 is able to increase their proliferation and expansion. So CD28 is not required for IL-2 to promote T-reg homeostasis. And this made us wonder whether IL-2 could substitute for CD28 in settings where co-stimulation was blocked. So here we're looking at Ki67 again in just normal Bauxi mice, and we can see that the Tregs are proliferating more than conventional T cells in the steady state. And this proliferation is blocked by antibodies to CD80 and CD86. But if IL-2 is provided at the same time, if the mice are injected with the IL-2 complex, as well as the blocking CD80 and CD80 antibodies, now the Treg homeostasis is maintained. And we don't really see a big difference in the conventional T cell compartment, probably because these cells are not expressing high levels of CD25, the high affinity IL-2 receptor. This is the collated data. So co-stimulation blockade results in a decrease in the percentage and absolute number of Tregs. But if IL-2 is also provided, then this decrease in Tregs is prevented. And there's not much of a change in terms of the conventional T cell counts. When we look at the phenotype of the Tregs, we can see that after co-stimulation blockade, Tregs have lower levels of CD25, but actually in the mice that received IL-2 at the same time as co-stimulation blockade, this doesn't happen. They have high levels of CD25, just like the mice that receive IL-2 alone. CTLA-4 expression is also downregulated after co-stimulation blockade. But again, this is prevented if IL-2 is also provided. And the collated data is just on the right. We don't see very much staining with TGF-beta, but this is decreased after co-stimulation blockade, but very nicely increased and maintained in the presence of IL-2. So IL-2 appears to be able to substitute for CD28 to some extent in maintaining Treg homeostasis and expression of effector molecules, at least in the steady state. So what about in the setting of autoimmunity, where you would imagine that now the conventional T cells are perhaps starting to upregulate CD25 expression? So to examine this, we looked in our spontaneous model of diabetes. So this is the same two transgenes, but now co-expressed in the same mouse. And in these animals, the ovospecific T cells respond to the ovalbumin that's expressed in the pancreatic islets. And there's a multicellular infiltration of the islets, destruction of the beta cells, and the mice develop diabetes with 100% penetrance. This is preceded by autoantibody production, and the T cells in this model show a follicular helper T cell phenotype. And in fact, we've also shown that this is the case in people with type 1 diabetes. And on that point, there's a potential added benefit of combining co-stimulation blockade and IL-2, in that by expanding Tregs that have high levels of CTLA-4, these should be capable of controlling TFH differentiation, as shown by very nice papers from Arlene Sharp and Shimon Sakaguchi's lab a few years ago. And IL-2 itself can directly inhibit TFH differentiation, so this may potentially be useful. So if we look in our autoimmune mice, now we've got a little bit more proliferation going on in the conventional T cell compartment. The proliferation of the Tregs here, we can see, is inhibited in the presence of the antibodies to CD80 and CD86. But if IL-2 is also provided, then Treg proliferation is maintained. But we're not seeing a big impact of the IL-2 on the conventional T cell compartment. And again, this is just collated data. So we have a decrease in the frequency and absolute number of Tregs after co-stimulation blockade, but this is prevented if IL-2 is also provided. So what does this mean then for blood glucose homeostasis in these animals? And to explore this, we adopted a two-phase treatment protocol, where during the first phase, we gave the animals co-stimulation blockade and IL-2 to try and inhibit the effector T cells but maintain the T-regs. And then in the second treatment phase, we took the co-stimulation blockade away and just kept the IL-2 to maintain the T-regs. And part of our thinking here was to actually allow the T-regs to use CTLA-4 to perform transendocytosis during this second phase. When we did tail bleeds at 7 weeks, we could see in the mice that had received co-stimulation blockade that T-reg frequencies were decreased as we expected, but this was not the case in the animals that had received the co-stimulation blockade and IL-2. And interestingly, this sparing of T-regs appeared to be selectively happening in the antigen-specific T-regs and not in the non-antigen-specific compartment. So in terms of blood glucose readings, the control animals in grey all developed diabetes. The mice treated with IL-2 or co-stimulation blockade alone had a delay in diabetes, as you would expect, but they still were all diabetic by 14 weeks post-treatment initiation, whereas the mice that had received co-stimulation blockade and IL-2 were highly protected from developing diabetes. And we were also able to delay treatment until the mice became dysglycemic, and at this point it's harder to control disease, but even so we still saw a marked protective effect of this combined treatment approach. And when we looked in the pancreatic islets by confocal microscopy, we could see that in the animals that received this combination therapy, there was still plenty of green CD45 positive cells infiltrating the islets, but we saw marked preservation of the insulin staining in red, and hopefully you can see that there are a lot of green FOXP3 positive T-regs within this infiltrate. So given these promising data from the mouse model, we've been interested in seeing if this same relationship between CD28 and IL-2 holds true in human cells. And in order to address this, we've used humanized mice. And these we've generated by injecting NSG mice, NOD, SCID, common gamma chain knockout mice with human CD34 positive cord blood cells. And these animals develop a human CD45 positive compartment that contains some CD4 T cells, and within those CD4 T cells we can see a fraction of FOXP3 positive T-regs. And these T-regs, as you would expect, have low levels of CD127 and have high levels of CD25 and CTLA-4. And of course, because we're now looking at human immune cells, it allows us to use more clinically relevant reagents. So we've used abatacept for co-stimulation blockade, and we collaborated with Roche to use an IL-2 molecule with an extended half-life. What we found is that the mouse treated with abatacept, the co-stimulation inhibitor, showed a decrease in the percentage and absolute number of T-regs, but if the IL-2 molecule was provided at the same time, then we didn't see this loss of T-regs, and we weren't seeing a huge effect in the conventional T-cell compartment with these interventions. When we look at the phenotype of the regulatory T cells, very similar to our work with mouse lymphocytes, we found that co-stimulation blockade decreased T-reg expression of CD25 and CTLA-4 and TGF-beta. But if IL-2 was provided at the same time as abatacept, these decreases were prevented, and the collated data is just shown over here on the right. So obviously there's a lot more that still needs to be done, but this suggests at least that the combination strategy may also be relevant to the human immune system. So just to summarize then what I've been talking about, we think that CTLA-4, CD28 and their shared ligands operate as an integrated functional unit. So it's not possible to consider perturbations in one of them without considering the impacts on the other players. CTLA-4 is a highly dynamic molecule. It's dynamically thresholding immune responses by restricting the availability of co-stimulatory ligands and restricting CD28 co-stimulation. And this type of regulation is highly quantitative and highly context dependent. It's very much influenced by the ratio of the target cell to the donor cell, the expression levels of all of the key molecules, the dwell time of the target cell on the donor cell, the CTLA-4 cycling rates, and so on. So there are many things about the microenvironment that can change how this regulation performs. And in practice, the CTLA-4 expressing cell is often a regulatory T cell, so t-reg homeostasis is very important for this. And it's clear that T-regs are indexed to conventional T cells via co-stimulatory ligands and IL-2. T-regs can use CTLA-4 to remove co-stimulatory ligands and decrease CD28 engagement and IL-2 production. And in turn, this availability of co-stimulatory ligand and IL-2 feeds back on T-reg homeostasis. And this fits very nicely with recent work from Ron Germain's group, modeling how IL-2 sensing instructs CTLA-4 dependent T-reg function. So co-stimulation blockade perturbs this feedback loop and impairs T-reg homeostasis, as well as impairing the effector T cells, which is of course what we would like it to do. But it may be possible to offset this by co-provision of IL-2 and spare T-regs and selectively suppress conventional T cell responses. So I will end there and thank everybody in my group, both past and present. In particular, I want to draw attention to Chenjing Wang, who is here and has led on the combination therapy project. And I'm enormously grateful to all of my funders and fantastic collaborators. And thank you very much for listening.
Lucy, that was, I didn't promise too much. That was a really fantastic talk. Thank you so very much for it. And as we do here at Global IMMUNOTALKS, I'm afraid we have to end quite abruptly. But before we log off, so first of all, my thanks to Lucy, our thanks to Lucy. If you have questions for Lucy Walker, please go and ask them on Twitter. Just look for Global IMMUNOTALKS and you'll be able to ask Lucy questions. And I'm sure there are plenty. Don't be shy about this. And as a reminder, next week, September 28th, Russell Vance will be our guest on Global IMMUNOTALKS. So thank you all for being here, and thank you, Lucy, in particular, for your wonderful presentation. Thank you.